Hello my friends, in this video we'll be taking a look at the autosomal DNA results, predicted phenotype traits, and G25 results of an Etruscan individual from Tarquinia in central Italy. So let's get into this individual's results. This individual is a male. He's carrying Y-DNA J2. I think it's J2B. Hold on, let's find that out. I know my trait predictor is displaying J as the individual's Y-DNA for the string, for the individual's Y-DNA string, but the only reason it's not displaying J2B is because he is not scoring anything for J2. No variants were found for J2, unfortunately, because of the low quality of the file. But if you look at the little spreadsheet over here, you will see that he's scoring 7 J2B variants out of 7 total, so he definitely has J2B. If only this file had any information for J2, if only this file had a little bit higher quality, he would be predicted for having J2B. So the reason he's not getting a prediction for J2B is because to score J2B, you need to first score J2, right? You need to score J2 in order to score J2B, and he's not scoring J2, unfortunately. So because of that, he's only scoring J. That's the string is breaking at J. That is the breakpoint. Because as you can see, he's got IJ. He's got 21 out of 21 variants for IJ. He's got J. He's got 48 out of 48 variants for J. But the string is broken when it comes to J2 because he's got 0 out of 0 total for J2. Unfortunately, the file does not contain enough variants for J2. And that is where there is breakage. And because of this breakage, even though he's got the variants for J2B, my program is not going to display J2B for the final string. And this is not something that you're going to encounter with commercial DNA files. Like if you have a MyHeritage or 23andMe or Ancestry DNA file, this is not going to be a problem because these commercial DNA files have all the relevant information for all the intermediate steps, like J2, for example. So you're not going to encounter errors like this with commercial DNA files. These types of errors can only be found with incomplete coverage files that result from conversions from BAM or FASTQ conversions, as is the case here. So this is very unfortunate, but still we can very clearly determine that his Y-DNA is J2B, which is actually a very Etruscan lineage. And this is an Etruscan individual. He's got an Etruscan lineage. Some information about the sample. He's from this location right here, Tarquinia in Italy. And he is from the first millennium BC, a little bit before the Common Era. His mitochondrial lineage is U5B2, which is a very typical mitochondrial lineage for Europeans. And in terms of the G25, the Europeans he's closest to with G25 are various North Italians. And what's very interesting to me as well is he's close to Swiss people and French people and even Romanians and Bulgarians. So which confused me a little bit because I was expecting this individual to be, a Etruscan, which are more southern, more western, and he seems to be a little bit more northern and eastern. So he has a little bit more Indo-European, a little bit more steppe admixture than what's typical for Etruscans in general. So he has a little bit more Indo-European related admixture than what's typical for Etruscans as a whole. He's even close to Romanians, and Romanians are much more northern than. I guess even Swiss and French are quite northern as well. So being close to Swiss and French is already evidence of being very northern for an Etruscan. All right, let's go ahead and check what he scores for Nashakot for phenotype. So it looks like he has hazel color eyes. Quite a great chance for brown eyes or blue eyes with a neighbor center as well. By the way, this is very light for somebody from central Italy. I think that a 2.7% odds for blue eyes and basically 19% odds for blue eyes with a neighbor center and 5.5% odds for green eyes. This is very great odds for light eye colors for somebody from this region and this time period. So compared to the rest of Etruscans, this individual is definitely very light. For hair color, he most likely has light brown hair. This is also extremely depigmented. This is the largest group. This is the largest category that he scores for hair color. So he most likely has light brown hair. Let that sink in. How many Central Italians do you know with light brown hair? I don't think I know that many. I don't know that many Italians in general, but out of the Italians that I do know, I don't know any with light brown hair at all. They all have either dark brown or black hair. So this individual is very light in terms of pigmentation. For skin color, likelihood distribution, it looks like he has palest or white skin, and in fact he's scoring higher for palest skin than for white. So he actually has palest. He's scoring the palest skin tone you can score with my trait predictor. So he has very pale skin, he has light brown hair, and he has hazel eyes. This individual is definitely very light in terms of pigmentation. Let's see his hair texture. For hair texture, he's scoring wavy or curly hair. And for nose shape, he definitely has a Greek shaped nose. So definitely more, I guess, longer, aquiline, leptorine shaped nose. Okay, for coloring related variants found in the file, looks like he has blue eye haplotype 2. Heterozygous for blue eye haplotype 2. That's very interesting. So he's got BH2, heterozygous for BH3, he's got BH1. Despite that, he's still scoring highest for hazel eyes, which is very, very interesting. Typically, when you have this kind of a genotype, you would be scoring blue eyes instead of hazel. With this type of genotype, you should be scoring blue eyes instead. So why is he scoring hazel eyes instead of blue when he's got this type of genotype? Let's find out. He's got 2 light color variants here. Okay, now I understand why he's scoring hazel eyes. He's got 0 light color variants in this version of SLC45A2. This is definitely very un-European. 0 light color variants here. This is also very un-European. So this makes sense. Now this makes sense. This definitely contributed greatly to him scoring the way he does. He does have another very interesting genotype in this variation of IRF4, which is very European and very hunter-gatherer, which greatly lightens the pigmentation of eyes, hair, and skin. But also there is these 2 genotypes in TRP1, which sort of darken the skin tone and hair. You have to go genotype by genotype and evaluate their impact, but I'm not going to do that. It's way too complex of a topic to get into. But essentially, the biggest impact in terms of why he has hazel eyes and not like blue or green is this right here. This is the biggest impact, the biggest reason for him scoring hazel eyes with my trait predictor instead of blue. The biggest impact in that regard is this genotype right here. But that's very interesting. So looks like he has hazel eye color. But if we look at this result with SNPR3, or if we look at this result with YSEC is going to say blue. But SNPR3, I'm very curious. SNPR3 is probably going to say blue as well. Let's check that out. I think SNPR3 is going to say blue for eye color as well. But let's check that out. Let's see. I think it's going to say blue. That's my assumption. Yeah, it says blue as well. Interesting. So, but it's not very precisely blue. It's saying 7 times more likely blue than brown, 38 times more likely blue than green and hazel. So it's not very precise for blue. It's kind of muddy, kind of all over the place. But SNPR3 is indeed saying blue for this individual's eye color as well. So it looks like my tool is out of SNPR3 and YSEC. My tool would be the one that's the outlier. That's gonna say hazel instead of blue. That's interesting. Okay, now let's go ahead and look at the Phenotype Oracle. And for the Phenotype Oracle, these are the phenotypes closest to this individual. So the closest phenotype is this. Very, very interesting. Corded phenotype with looks like hazel eyes, looks like brown hair color, and I guess palest skin. I guess it does fit the prediction for him. I don't really have a phenotype that fits the prediction to a T that would have like really, really pale skin and light brown hair and hazel eyes, but this does fit it as close as it gets. The nose shape fits. I guess the curly hair doesn't really have curly hair, so the hair texture doesn't really fit. The second closest phenotype, I guess it also sort of fits as well. The third closest phenotype, I guess it also fits sort of, but I don't think any of these phenotypes have curly hair. Now let's look at the mixtures, the Phenotype Oracle. So it looks like the closest mixtures is a mixture of 50% this plus 50% that. I don't see any of them having curly hair. Looks like this one on the very bottom, on the very right. The bottom one has curly hair. Okay. Well, I guess this phenotype right here is sort of curly-ish hair. I don't know. I don't remember what I put down for their hair texture. because all of these phenotypes, the way I did them is I did like a hash map, and they all have certain values attached to them that represent their hair texture, their nose shape, their skin color, their eye color, their hair color, and all these phenotypes have certain values attached to these attributes. So all of them have a certain hair texture attached to them, and I don't remember what I put down for the hair texture of these phenotypes exactly. So some of these may be like slightly curly, so I don't remember what exactly they are, but that's very interesting. So this one on the bottom though, this one is definitely curly, because you look at it, it looks a little bit curly. I made sure that I put some phenotypes with curly hair in there in the hash map. All right, so looks like pretty interesting. So they all kind of look the same, to be honest. All these phenotypes kind of look the same, except for like this one. This one looks a little bit ethnic, and here looks a little bit ethnic. All right, let's close that. So this individual is pretty white, definitely quite a European-looking guy. Let's look at the biomarkers panel. So for the biomarkers panel, he's got a predisposition to higher levels of vitamin D, which is good. A lower level of LDL cholesterol, which is really good. A lower level of HDL cholesterol, which is okay. Typically, you want to have higher level of HDL and lower level of LDL. So this significantly lower level of LDL cholesterol is really good to see. He's got a slightly higher level of glucose, which is okay. Slightly higher level of hemoglobin, which is okay. Lower blood pressure, which is really good to see. So definitely predisposed to lower blood pressure. Higher level of iron in the blood. So it looks like he does carry hemochromatosis variants. And this is actually a crazy score. 134, that's a crazy score. I don't remember what the units stand for, but let's look up hemochromatosis. So he's got 2 copies of the H63Z variant. Yeah. So yeah, that's a crazy score for hemochromatosis. That makes sense. That totally makes sense. So he's got definitely some predisposition to hemochromatosis, and you can see that from the biomarkers panel. For sex hormone binding globulin, he's got lower odds for that. I don't even remember what that's implicated in. For red blood cell count, he's got lower odds for that. For telomere length, very high number of base pairs for telomere length, so he's definitely predisposed to longer lifespan. Very good to see. And slightly shorter height for expected height. Okay. Italians aren't really known to be very tall, so, but I don't know how much Etruscan ancestry do Italians really have. I don't know. Okay, let's go ahead and look at the polygenic risk scores. I am going to ignore everything that is average and only look at the stuff that is either very above or below average. So it looks like he has a very high score for kidney stones, a very high score for hemoglobin E disease, low score for migraine, high score for lupus, very high score for gout. It reminds me of that meme. Hold on, let's look it up. I have gout tattoo. Yeah, it reminds me of this meme. Tattoo ID if you have 11 fingers and gout. Yeah, I saw this on Reddit a couple days ago. It's pretty funny. Okay, let's get back to the topic. Gout is like when you have too much acid in your extremities, and it ends up causing your bones to collapse. Looks like he has below averages for eczema. Looks like he has below averages for exfoliation glaucoma. Looks like he has below averages for cataracts. Very high odds for age-related macular degeneration. Okay, we're not going to look that up because there is no point. He has above averages for rheumatoid arthritis. He has below averages for Tourette's. He has. So we remember that the highest risk scores we've seen thus far was AMD and rheumatoid arthritis. He has above averages for epilepsy. He has above averages for asthma, above average for leukemia, above average for vitiligo, above average for a lot of stuff. So he's definitely got a lot of predispositions that are unhealthy. Above average for myopia. Yeah, this individual has a lot of predispositions that are really bad. Like sometimes when I go through these samples and I see that they have below average for everything. So in this case, I'm just saying above average for pretty much everything. So I don't even know what to talk about because there's so much above average predispositions to stuff. Above average for corneal astigmatism. Below average for stroke. Finally, something below average. Above average for male pattern hair loss. So he's predisposed to stuff like balding, losing hair, below average for atrial fibrillation, good to see, below average for deep vein thrombosis, below average for ADHD, above average for depression, below average for bipolar type 1, below average for schizophrenia, below average for type 2 diabetes, and above average for type 1 diabetes. Wow, that's interesting. So type 1 diabetes, I definitely want to see the score for HLA gene panel, because this genotype for type 1 diabetes is very interesting. I think I just answered my own question, because 7.6 times the averages for multiple sclerosis, he definitely has a bad score for the HLA gene panel. Yep, that makes sense. And 3.6 times the average for Alzheimer's. So I feel like there is going to be the score for the HLA gene panel is going to be really bad, and there's a lot of predispositions to autoimmune disorders. Now let's scroll up a bit and see the score for asthma and lupus. I think I skipped over that, but he's scoring high for lupus. And what is he scoring for asthma? And he's scoring high for asthma as well. Yes, so I think the score for the HLA gene panel is going to be quite bad. That's what I think. Overall, he has a high predisposition to various autoimmune conditions. That's what it is. That's the problem here. Okay. Let's see for the cancer section. No risk variance for breast cancer. No risk variance for breast cancer at all. Now that's good. So he's quite protected from breast cancer. That's really good to see. That's rare. 0 out of 18 for breast cancer. That's really good to see. Only 4 risk variance for testicular cancer out of 12. That's also really good to see. But he's got 2 risk variance here in Keto G. So that's okay. I guess it's not so bad. 0 risk variance for celiac disease out of 6. That's okay. No risk variance for GSS. 4 risk variance out of 20 for Crohn's. That's okay. And we don't see any other rare conditions that he's predisposed to. Porphyria, 2 out of 14 total. 2 risk variants that are fairly common. That's okay. And we don't really see... Whoa. Okay, whoa. He's got 1 risk variant for Waardenburg syndrome. That's crazy. But I think this might be a miscall. 6 risk variants for allergies out of 20 total. Okay, pretty cool. 1 risk variant for familial hypertrophic cardiomyopathy. But this probably is a miscall as well. And we don't see anything else. Alright, so what I'm concerned about is the type 1 diabetes, multiple sclerosis, and everything relating to HLA gene. Let's go over the monogenic traits and find out what we see here. So it looks like for warrior versus warrior phenotype panel, he's definitely more predisposed to warrior phenotype, so slower dopamine reuptake and higher dopamine level. So if you take into account COMT, MAOA, and MAOB genotypes, if you take all of them together into account, then he's more predisposed to being a warrior. Although here on this panel looks like there is no genotypes in MAOB. It's just COMT and MAOA. Still though, he's scoring 50% for odds of warrior. For DRD2, he's scoring intermediate number of dopamine D2 receptor sites in the brain, so intermediate predisposition to such conditions as schizophrenia and bipolar. And for levels of 5-HT and serotonin, he's scoring some predisposition to lower or intermediate levels of 5-HT and serotonin. So some very slight predisposition to such conditions as anxiety or depression. He does not have dwarfism. He's not a dwarf. And he does not have any predisposition to sleep disorders. Pretty good to see. Okay. For autism, it looks like he has predisposition to lower odds of autism. So that's pretty good to see. Not really autistic. For lactose persistence, let's look at that. Looks like he has predisposition to higher or intermediate loads of lactose intolerance. So he's got some predisposition to being lactose intolerant. Based on these 2 SNPs, which isn't that much, I suspect that there is like hundreds of different SNPs that play a role in lactose persistence. I was doing a lot of research and I couldn't really find those hundreds of SNPs. Looks like people haven't really been conducting research into that topic. I could only find 4, so my trait predictor looks at 4, but he doesn't have any variants for lactose persistence. So based on that, what can we say? He's probably lactose intolerant, but then again, a lot of people don't have any variants for lactose persistence, and they still are lactose persistent. So because of that, my trait predictor is very liberal. It's very tolerant, and you can score even if you don't have any variants for European lactose persistence, you will still score, mostly you will score intermediate. That's just how my program works. It's very, very, very tolerant to people not having any variants for lactose persistence, because I know the way it works is that most people in the world don't have any variants for European lactose persistence, and most people in the world are not lactose intolerant. So I take that into account when I built my program. I took that into account completely, and I made it very, very tolerant of that fact. And even if you don't have any variants for that at all, it will still predict you to be intermediate. It's not going to predict you to be lactose persistent, but it's going to predict you to have intermediate odds of lactose persistence. That's the way it works. And that's why this individual, despite not having any variants for European lactose persistence whatsoever, is still scoring intermediate odds of lactose persistence. That's just the way my program works. And this is actually the only trait where I made it this way. Every other trait it's not this tolerant at all. This is the only trait where I decided to make it work this way. For OXTR and empathy gene, he's scoring a higher level of empathy based on these 3 SNPs. For diabetes, let's see. He's got... I don't want to go over this, to be honest. Okay, for Alzheimer's, I remember he was scoring very high for odds of Alzheimer's. That is because he has a risk variant for Alzheimer's in this APOE variation. So he actually has a risk variant for Alzheimer's in APOE. That's very interesting. For multiple sclerosis, he is HLA homozygote. Wow. Okay, wow. So he has risk variants for multiple sclerosis in the HLA gene, and here he's homozygote. Here he's a carrier. Here is also homozygote. I should have specified, is this a homozygotic or is it a heterozygotic genotype? I should have specified that in my program, so I can't really know if he's a carrier or if he's a homozygote. Probably this is a heterozygotic genotype, but I should have specified that in my program. Okay, that's very interesting. Very, very fascinating. For cardiovascular disease panel, he's going to score intermediate or lower odds of cardiovascular issues. So this is going to really screw up the HLA gene panel. The HLA gene panel is going to look horrible because of this. And for vitiligo panel, he's also scoring risk variants for the HLA gene. Wow, this might be the worst. This individual might have the worst HLA gene result that I've seen in a long, long time. Just based on what I've seen so far, this might be the worst result for HLA gene panel that I've seen in a very long time. For facial morphology panel, European genotype and EDAR, and 2 genotypes for slightly thinner eyebrows, larger nose size, a more upturned nose, nose pointing up. Very interesting. He does not have micropenis, really good to see. He's got a mix of muscle types, slightly more sprinter rather than endurance athlete. He's got photic sneeze reflex. He's not an Asian flusher, and he's got lower odds of alcoholism. Okay. He's got lower odds of cannabis-induced psychosis. That's a very European trait to have. He's got no risk variants for pain hypersensitivity out of 8 total. Really good to see. Let's see what else. He is not albino. He doesn't carry any variants for albinism. He also does not carry any variants for familiar Mediterranean fever. He has below average odds for alcoholism. Very cool. So he's not predisposed to being an alcoholic. He's got genotype for normal homocysteine levels in MTHFR, so homocysteine panel is probably going to look good. I cannot say that about the HLA gene panel. HLA gene panel is probably going to look very, very, very horrible based on what I've seen so far. Okay, leukemia panel, he's got some variants for... Yeah, he's got a lot of risk variants for leukemia, actually. So the important risk variants are these two, and he's got risk variants for leukemia in both of them. These are not as important. And we remember he was scoring higher for odds of leukemia as well. So he's quite cooked when it comes to leukemia risk variants. For rare diseases and traits panel, let's see what he has got here. So far, what we remember about the sample is he's very predisposed to various autoimmune conditions, such as, type 1 diabetes, celiac disease, multiple sclerosis, stuff like that. He's very predisposed to such conditions as this. Addison's disease. Let's see. I'm not seeing any rare conditions here. Okay, he has this genotype right here, which leads to slightly increased risk for several autoimmune diseases, including Addison's disease. Okay, and this genotype leads to higher odds of eosinophilic esophagitis. But that's the only thing I see here. I don't see any other conditions that he's predisposed to. So really, he's quite good in terms of that. But the HLA gene panel is going to look really horrible. He does not have increased risk of celiac disease and lupus here. He doesn't have any HLA DRB1 alleles, which is really good to see. So this is going to affect the HLA gene panel in a good way. And he doesn't have any risk variants for Canavan syndrome, really good to see. And now let's go to the HLA gene panel. And for the HLA gene panel, he's actually scoring highest odds of autoimmune disease. Wow, this is crazy. So this is pretty much what I was expecting, but I just wasn't prepared to see this in the result. So he's definitely very predisposed to several autoimmune conditions based on what we see here. Highest odds of autoimmune disease. And we see what contributed to this score. It's the risk variance for vitiligo. It's the risk variance for multiple sclerosis. This contributed to the score in a positive way, but there is also this, which leads to higher odds of type 1 diabetes. And there is also this, which leads to... This actually contributed to the score in a positive way as well. Lower odds of allergies. But overall, this is a very negative score. And this is something I'm probably going to put in the thumbnail, actually. So this individual is very much predisposed to various autoimmune conditions based on what we see here on the HLA gene panel. All right. For the MTRR gene, looks like he has no risk variance for disorders of intracellular colon metabolism. Really good to see. And for homocysteine panel, looks like he has intermediate homocysteine levels. Really good to see. All right. For muscular dystrophy myopathies, no risk variance for that. And he's got 2 risk variants for adrenoleukodystrophy out of 24 total. That's really crazy. I cannot confidently say that this is a miscall. The thing is, I can't say that because it could be a homozygous genotype. It can only be if it's legit, it can only be a homozygous genotype because it's on an X, and this is a male that I'm analyzing, so it can only be a homozygous genotype if it's on an X, and I'm analyzing a male. But also, I don't really believe it too much. I don't really trust it too much because it's just such a rare condition, and all of these variants are so rare and uncommon. And also, this individual would have died at a very early age if he actually had this condition. So I don't know if I think I'm gonna chalk that up to miscalls as well. I'm gonna go ahead and chalk that up to miscalls. Let's look this up. Is this rare? Is this common? It might be common. Yeah, this is common. So this is probably not a miscall. This genotype, the 1 risk allele here is probably not a miscall. But the risk variance for adrenoleukodystrophy, this I think is a miscall actually. For color blindness panel, no risk variance for color blindness. Really good to see. For FTO gene panel, looks like he has a predisposition to lower or intermediate odds of obesity. Really good to see. So based on these 1, 2, 3, 4, 5 SNPs, he has lower predisposition to obesity. For syncope panel, based on 6 SNPs, he's got lower odds of syncope. And for bio traits panel, let's see. What can we talk about here? Less able to detect beta iodine in floral fragrance. So in more simple terms, can't smell flowers. Normal or smellier body odor, normal colostrum, typical for non-East Asians. So he's smellier. East Asians tend to be less smelly. That's pretty much it. I don't want to talk about too much else in this file. For blood group panel, it looks like his blood type is actually type A. Very interesting blood type. I don't see it very often with the samples I analyze, but looks like this individual has blood type A. Very, very cool. Based on 1, 2, 3, 4, 5, 6 SNPs, his blood type is predicted to be type A. Actually, I say 6 SNPs, but there's a couple more SNPs that go into the calculation that aren't displayed on the board. It's like 1 or 2 SNPs that I haven't made showable. They don't always show up, that's why I don't mention them. But there's like 1 or 2 additional SNPs that play a role in the calculation that don't show up on the board. I don't mention them because they aren't always present. But yeah, so they may or may not be present in this case for the calculation. But in this case, in this individual, he's most likely type A for the blood type. Very, very interesting. And that's pretty much it. Well, thanks for watching my video until the very end. I hope you were satisfied and entertained. I hope you liked it. Leave a like and maybe subscribe if this is the first video you watch from my channel. Make sure you read the description of the video. In the description, you will find the links to download this file in 23andMe format. Make sure you check out my software. Make sure you check out my itch page where I sell my Trait Predictor. Trait Predictor is the software that you can use to generate a trait report, such as what you've seen in this video. This report will work for MyHeritage, 23andMe, or Ancestry raw DNA files. And also, you can find in the description of the video a link to purchase a single report, not the executable, but just a single report, and that's going to cost you $4. So you just upload your file to me, and in a couple minutes, maybe a couple hours, it's never going to take me longer than a couple hours, I'm going to send you back your report with Trait Predictor. It's going to look something like this, something like that, and you can explore it, and that's going to cost you only $4. Well, anyway, thanks for watching. Make sure you check out the description of the video, and goodbye.